Hey everyone, welcome back to this session. Today we are going to discuss about a question of recombinant DNA technology which have been asked in previous year question of CSIR LifeScience NET. So let us have a look with this question. This is a question. First of all, before reading the question, we have to understand what it is asking. Here through the option we can see that it is asking about the base pairs which we are getting after the cloning. Okay. Now see, let us read the question. A researcher attempt to clone a 630 base pair coding sequence of a gene downstream to a bacterial promoter which is of 500 base pair in length. See what the researcher is going to do. It is going to clone a gene which is 630 base pair. Now see, this is 630 base pair. It is going to clone a gene. The gene which is needed to be cloned is a 630 base pair. See, this is a gene. It is of 630 base pair. Where it is going to be inserted? It is going to be inserted downstream to a bacterial promoter which is of 500 base pair in length. This bacterial promoter is 500 base pair in length. Downstream to this promoter, this gene is need to be inserted. Let us see what it is telling. For overexpression and purification of the encoded protein, the gene sequence was isolated as a small fragment. Recognition sequence CCC arrow GGG. Arrow indicates the site of restriction and cloned at a SMAI site located downstream to the promoter. That means the gene which is needed to be inserted, it will insert in the SMAI site which is located downstream to the promoter. After promoter, it is located. The SMAI, look here, the SMAI is located downstream to the promoter region. Okay. Now this gene will be inserted in this region. Now let us see what it is telling. The gene sequence contained a single site for EcoR1. That means the gene which is needed to be inserted, it is having a site of EcoR1. Let us see where it is having. Located 30 base pair downstream to the start codon. EcoR1 is present. Here we have a site of EcoR1. Where? After 30 base pair downstream to the start codon. Here in this gene, the start codon is this one, and after 30 base pair, the EcoR1 site is present. So, the total gene was of 630 base pair, of which the first part up to 30 base pair, after 30 base pair we are having a EcoR1 site. So, the remaining part will be 600 because 30 is for EcoR1. So, the remaining one is 600 base pair. This gene is to be inserted in SMAI region of the bacteria. Now, let us see. A schematic representation of the plasmid along the location of some restriction enzyme site of vector is given below. Now, the researchers screened the obtained colonies by a double digestion using HindIII and EcoR1. What happens after the insertion of this gene into the vector? What the researcher did? He did double digestion using HindIII as well as EcoR1. Now, first of all, let us draw this diagram. Here we are having, this is the total part. Here we are having HindIII. Here we are having the region of SMAI where the gene is to be inserted. Then we are having a site known as EcoR1, and this is the promoter region. Promoter region is 500 base pair, and the gene will be inserted in this region. In this region the gene will be inserted. This is the part of the gene. Okay. When the gene will be inserted, the SMA1 will be no more. So we are cutting this. Now the gene is having 30 base, after 30 base pair of the gene it is having echo R1. Let us draw here. This is a total gene, this is a total gene which is inserted, and here we are having a region of echo R1 and it is having 30 base pair. So the remaining part will be 600 base pair, it is of 600 base pair. What the researcher did, it made double digestion using hint 3. First was hint 3. So the cut was made here in the hint 3 region. Another cut was made in echo R1. We are having in the two regions we are having echo R1. So in the two regions the cut will be made. Here it is the first region where the echo R1 is present. Here one cut will be there, and in the last segment there will be another cut. So what are the two fragments which we are getting after the cut? We have to explain that. So after the cut, here we are having 500 base pair plus 30 base pair, that is we are having a total of 530 base pair. How? Because a cut was made here which is having 500 base pair, another cut was made here in the echo R1 side which is of 30 base pair. So after the two cut we will get a segment which will be of 500 plus 300, that is 530. Another cut is made in the echo R1 side which is in the bacteria. So one cut was made here, another cut is here. So we are getting another fragment which is of 600 base pair. Okay. Now in the question it is asking which of the following restriction digestion pattern would represent the restriction profile of the desired clone that could be used for overexpression. In the first option it has been given that 530 base pair plus 600 base pair plus vector backbone. Yes, this is our correct answer because we have got here 530 base pair, another one is 600 base pair, then we will getting a vector backbone. Okay. Thank you for watching this video till the end. If you have understood this question, please do subscribe to my channel and please share this video to everyone those who need. Thank you.